Deparkar, welcome you to this webinar on Carbetocin in action. Doctor, as you know, at Heathrow, gynecology, orthopedics, and pulmonology are very strong multiple therapy areas for us. With a strong R&D support, we have developed many unique formulations in keeping patient benefit in mind. Our endeavor is to develop products and formulation combinations which will improve patient compliance and ultimately clinical outcome. We have developed NSAID patches, reverse treatment patches for patient compliance. At Friends or Division Doctors, gynecology obstetrics IVF is an important therapy segment for us. We market products for vaginal health like Feminora, Pelletra, etc. We have formulated and introduced a vaginal film containing lactic acid. In oral formulations, besides dydrogesterone and other products, we market Milotin, a unique nutraceutical for PCOS management. Milotin contains myoinositol, D-chiroinositol, along with chromium picolinate and melatonin. Recently, we also introduced Carbiact, our brand of Carbetocin. This Carbetocin we know is used for the management of PPH. PPH we know is a condition wherein we have more of complication and unexpected condition, expect the unexpected as they say, and is not always welcome. To speak more about prevention and treatment of PPH, we have with us renowned speakers, Dr. Bedi and Dr. Misha Gupta. Welcome, madam. Dr. Bedi is MBBS DG or DNP. Madam is Professor of Obstetric Gynecology in Adesh Institute of Medical Science and Research Institute, Bhatinda. Founder Secretary of Bhatinda Menopause Society, member of FOXI AMSI. Madam has various publications in national and international journals and a reviewer to many journals. Dr. Misha Gupta is Assistant Professor, Adesh Institute of Medical Science and Research Center, Bhatinda. Worked as an attending consultant at SPS Apollo, Ludhiana. Madam is trained in human embryology and andrology and has performed procedures like oocyte retrieval, cyst aspiration under sonography guidance, and embryo transfer. Has interest in management of high-risk obstetrics of all patients conceiving through infertility, through treatment for infertility, including management of complications like OHSS, ectopic pregnancies, and fetal reduction of multiple pregnancies. Welcome, Dr. Bedi. Welcome, Dr. Misha Gupta. Handing over the session to you. Thank you.
Thank you, sir. Good evening, everyone. I would like to thank my teacher, my guide, Dr. Bedi ma'am, and respected organizers of Hetero Healthcare for giving me this opportunity to put my views on this topic for comparison of carbetocin versus oxytocin in postpartum hemorrhage patients. We have been planning since long to conduct this study at our institution. So finally, we did it on 80 patients. 40 patients were enrolled for carbetocin and 40 for oxytocin.
Priyanka, can you share the slides with us, please? Yeah, sure, ma'am.
Thank you, Priyanka. First is the introduction part. Postpartum hemorrhage is the major cause of morbidity and mortality. If it is within the first 24 hours after delivery, it is primary PPH. When PPH is occurring between 24 hours and 12 weeks postnatally after delivery, it is secondary PPH. It may result from failure of uterus to contract adequately, genital tract trauma, retained placental tissue, or maternal bleeding disorders. Uterine atony is the most common cause and consequently the leading cause of maternal mortality worldwide. Next slide. Incidence of PPH is reported to be 2 to 4% after vaginal delivery and 6% after cesarean sections. In India, sample registration scheme, PPH accounts 38% of the maternal deaths. Next slide. Currently, oxytocin is the first choice uterotonic for the active management of third stage of labor, recommended by WHO to prevent postpartum blood loss. Prophylactic use of oxytocin after delivery of the baby has shown to reduce the incidence of PPH by about 60%. It has half-life of approximately 3 to 4 minutes and duration of action is 20 minutes. Whereas carbetocin is a long-acting synthetic agonist analog of human oxytocin. It has longer half-life of 40 minutes and elicits prolonged uterine response up to 1 hour after a single dose. Its prolonged uterine activity may theoretically offer advantages over oxytocin in the management of third stage of labor, as it is not required to be administered as an infusion, unlike oxytocin. Its greatest advantage is it is heat stable and can be stored at room temperature. It has proven to retain its efficacy when stored at 30 degrees for about 36 months. This is particularly relevant in a country like India, where cold chain maintenance is a challenge. Aims and objectives of a study aims to compare the effect of carbetocin with oxytocin in prevention of PPH in term patients after vaginal delivery. Objectives: to observe the effect of carbetocin in comparison to oxytocin for the prevention of blood loss in patients with vaginal deliveries. Second, to observe the use of additional uterotonic agents after administering carbetocin or oxytocin in vaginal deliveries. Materials and methods: we enrolled 80 women with term pregnancies giving vaginal birth and compared the effect of heat stable carbetocin at a dose of 100 micrograms IM with oxytocin as at a dose of 10 international units IM administered within 1 minute after vaginal delivery by quantification of blood loss with either of the two drugs. Quantification of blood loss was done by a list of dry weights for delivery items that may become blood soaked with directions on how to calculate blood loss was created. The weight was hence subtracted from the wet weight and amount of blood loss was calculated. Second, the total volume of fluid collected in the under buttocks drape was measured. Third, the pre-placental fluid volume was subtracted from the post-placental fluid volume, keeping in mind that most of the fluid collected after the birth of the placenta was blood. Inclusion criteria: primary gravida, multigravida, gestational age 37 to 41 weeks, age of the women 20 to 35 years, singleton pregnancies. Exclusion criteria: cesarean sections, multifetal pregnancies, women who have allergies from either of the drugs, uterine surgeries like myomectomy, women who have cardiovascular, renal, or hepatic disorders, any maternal or fetal high risk. Results: the mean gestational age in both the groups was around 38 plus weeks, with average age of the patient 25 to 26 years, which are very much comparable and statistically significant. The mean blood loss in oxytocin group was 512 ml and other one was 456 ml, which was found to be significant. However, the mean pre-delivery HB and post-delivery HB in both the groups were statistically insignificant. Next slide. Over to Mini ma'am.
Just the previous slide, Priyanka. Yes. So the most important thing which our study came out with was that in the oxytocin group and the carbetocin group, when we compared the number of extra uterotonics which were required after the delivery of the patient, despite giving oxytocin or carbetocin in the group, the number of additional uterotonics which were required were a little higher as in the oxytocin group as compared to the carbetocin group. So this was found to be statistically significant. That's saying an additional uterotonic, which was in the form of maybe a misoprost or a carboprost, which we required to save PTH. It was more in the oxytocin group as compared to the carbetocin group. So in this study, actually, we attempted to compare the effect of carbetocin versus oxytocin in the active management of third stage of labor. We took only vaginal delivery patients. However, the study is still ongoing, and now we will be comparing the cesarean section patients as well. But what we found was administration of IM 100 micrograms of heat stable carbetocin was not anyhow inferior to the administration of IM 10 units oxytocin on the basis of the primary outcome, that is blood loss. Right? So blood loss in carbetocin group was almost less as compared to the oxytocin group, and it was significant. The p-value came out to be 0.028. The need of additional nutrotonics, it was again significantly lower. This was our secondary objective. So this was again significantly lower in patients with the carbetocin group as compared to the oxytocin group. And again, the p-value was 0.025. And obviously, a good safety profile is a key consideration for any nutrotonic agent. The safety profile of both the drugs was also found to be comparable in our study. We had no incidence of any adverse effects in either of the group. I mean, not only the oxytocin group, even in the carbetocin group, we did not find any adverse effects in the first 24 hours post-administration. Next slide, please. So compared with oxytocin, if we talk of heat-stable carbetocin, it does not need cold chain transport and storage. What I personally feel is this is the best part India needs to take care of. Therefore, it is convenient to be stored in facilities at room temperature in low and middle income countries like ours, where cold chain transport and storage is not available. Nevertheless, there's a huge disparity in the price between oxytocin and the carbetocin, and so health policies may take time to compare it further. So would you like to take up the questions, please? Okay, so there have been... Yes, Priyanka, you take up the questions? Dr. Prashant?
Dr. Prashant, sir.
Can you hear me? Can you hear me? Hello?
Yes, sir.
Yeah, sir.
My question is, in which of the cases you would prefer carbetocin or oxytocin for prevention of PPH?
So first is the cases where carbetocin can be used. Basically, it can be used in any patient, any random patient can use carbetocin. Only we need to rule out as heart disease, preeclampsia, eclampsia, maybe all hypertensive disorders of pregnancies.
Can it be used in...
Second one is, can it be used in retained placenta? Literature is not available, but better to avoid carbetocin as a routine use in retained placenta.
Can it be used in Rh negative mothers?
Yes, it can be used in Rh negative pregnancies.
Does the PPH intensity vary, madam? This is for my interest, in Rh negative and Rh positive, or is it almost the same?
Should I take it up?
Does the intensity change because of Rh positive and Rh negative? Intensity of bleeding, I mean.
It is actually not about the intensity. So what we are considering in Rh negative mother is a strong contraction may lead to isoimmunization. Like it may lead to a little bit of Rh positive blood going back into the mother,
Okay,
and that may cause isoimmunization or sensitization for the
Immunization,
next
yes,
3 months.
correct.
So, as far as the nutrotonic effect of carbetocin is considered, it is almost equal or similar to oxytocin. If
Oxytocin
we can use oxytocin safely, perhaps we can use carbetocin as well.
as well.
Although there have been no side effects till now labeled as far as carbetocin may not be used in Rh negative mothers, there's nothing like that.
The patient profiles where doctors can anticipate PPH and use carbetocin straight away for prevention of PPH. What are those patients' profile? Apart from uterine atonia, of course, I mean, it's a general condition.
Yeah, look, the best part about obstetrics is that we need to be prepared. Unless we anticipate a problem, definitely we will not be able to handle it. So there are a few cases where you should anticipate that this patient may go into PPH, and that is where we should be prepared for it. So any overdistension of the uterus, maybe multiple pregnancy or polyhydramnios, or a patient who has been for a very long time, she has been in the second stage of labor, or she has been in the first stage of labor, has been induced for a very long time. Any severe anemia, anemic patient, maybe some SOLs in the uterus, maybe fibroid and all in the uterus. All such cases, we do anticipate PPH, and there is nothing like that we cannot use carbetocin. Only thing is, one thing I would like to mention is that there are cases with preeclampsia, we anticipate PPH in these patients as well, but carbetocin is guarded in such a patient.
Can carbetocin be used for prevention of PPH if oxytocin was used in second stage?
Sorry?
Can carbetocin be used for prevention of PPH if oxytocin was used in second stage?
Yeah, 100%. We can do it. We can do it. There's nothing like that if oxytocin was used in first stage of labor or during induction or augmentation, we cannot use carbetocin. There's nothing like that. We can do it. No issues at all.
If the patient is not responding to oxytocin, looking at the receptors, that both of them will act on similar receptors. If the patient is not responding to oxytocin, would she respond to carbetocin?
It has been seen that they do respond. Obviously, theoretically, it feels as if the same receptors, the carbetocin and oxytocin, are supposed to work. But it has been clinically seen that if, despite the use of oxytocin, you give carbetocin, it definitely works and PPH is very well controlled.
Maybe there are a few receptors. Yeah. If patient is not responding adequately to carbetocin, what additional ureotonics can be used or should be used? Yes, if patient is not responding to carbetocin, what other additional ureotonics should be used?
Oh, sorry, sir. Sir, prostaglandins can be used. Misoprostol, carboprost can be used.
Okay. Looking at temperature variation in our country, I suppose that's the greatest plus for carbetocin because it can be stored under for 30 degrees. You don't require, I suppose, because
Yes.
electricity is an issue, and looking at the interiors, it will still be an issue everywhere.
Yes, sir.
And in your the case study that you described, all patients were vaginal delivery. But what is your experience on carbetocin in cesarean sections and in twin pregnancies?
We have used carbetocin.
Madam, you're mute. You're on mute. You're on mute, madam. Madam, you're on mute.
I'm sorry.
Mute.
I'm sorry.
Okay.
What study we have done till now was about only vaginal deliveries with 80 patients, and we had wonderful results with carbetocin as well. But then the study has yet not finished. We are taking it further. We are putting it further now for cesarean sections. Although we are getting very good results, I mean, we have not yet had even a single patient who landed up in the PH, but we are still to bring up the results and publish them.
Correct. And retention of fluid, ADH effect with carbetocin, how intense it is?
No, we have given hardly one injection of carbetocin with this, no? So hardly we have encountered any side effect, any fluid retention, any problems with the patient till now. Not even a single case of any side effects we have mentioned till now.
Even in twin pregnancies, I suppose this would be a wonderful drug because of uterine atony.
It could, absolutely, sir. I mean, we did not get even a single patient of any side effects. Our study was ongoing. We were regularly monitored whether we had any side effect, we had any adverse reactions with the drugs. We did not have even a single. Very happy about it. The safety profile is wonderful.
That is a very interesting question. Is carbetocin a reserve drug or should be used as a first line treatment for PPH?
The problem is with carbetocin, I'll tell you, sir. Even though the safety profile, the usage, the ease of use, everything is wonderful with carbetocin. The only issue, what I personally feel, would be the cost. Correct. The cost of one injection of carbetocin as compared to cost of 10 units of oxytocin. So, if we talk of governmental policies or health policies, right? I mean, the health policy makers need to understand that we are not spending 10 rupees for oxytocin or 500 rupees for carbetocin. I mean, taking the rough charges. But it is actually with oxytocin, we are putting in a cost of that cold storage as well, which may be maintained, which may not be
Absolutely.
maintained. And the
Yes.
patient has to face the brunt of it. If the oxytocin is good enough, nicely saved, nicely stored, and then given to the patient in time with that
Correct.
cool effect, only
Correct.
then the patient can be saved, right? So
Correct.
if we think of that extra cold chain charges, obviously oxytocin is very costly as compared to carbetocin. But
And again,
it still needs to come up
yeah.
in health issues, right?
And again, with oxytocin, you require a IV device, IV, I mean, a setup, IV bottles, IV trip you require.
Yeah, everything,
Unlike,
everything,
yes,
right.
unlike carbetocin.
Unlike
Whereas,
carbetocin, exactly. The
yes.
ease of administration is also
Yes,
wonderful with
yes,
carbetocin.
yes. So cost aside, otherwise carbetocin should be used as a first line. I mean, it is good for patient because we know
Obviously,
PPH,
even in
yes.
our, I mean, the study which we did, the conclusion also we concluded as if this should be considered as a first line drug for prevention of PPH.
Correct.
Although the health ministry still has to take a call on
Yeah,
it.
that is a different thing. Efficacy, safety wise, tolerability wise, carbetocin and other uterotoxins are almost same. Or carbetocin is
Absolutely
same.
same. Absolutely same. In fact, if we talk of efficacy, I think this is better. The use of additional uterotonics can tell us we had to use additional uterotonics more in the number of oxytocin group rather than as compared to the carbetocin group. So if we take up the efficacy, this is better, undoubtedly better.
And unlike prostaglandins or other drugs, this will have a lesser side effects. Yes,
That we
this will
take,
have
that is why we take them
side
as the
effects.
second line, like we do not put it as the first line. That is
Correct,
right.
correct. I think that is all, madam. I'll see something else. Thank you, all, madam. I think that's all. Thanks for your wonderful
That was really
insights on
nice,
yes.
Dr. Prashant, to have you. I hope
Thank
the
you, sir.
discussion was good enough and it was knowledgeable for everybody.
Yes. And I thank all the doctors who attended this session, all the doctors who participated, both of you. I thank the field personnel of Hetero for super coordination. I also thank marketing team Hetero for conceptualizing this. And once again, thanks all of you. Signing off. Thanks, Dr. Mishi, and thanks,
Thank you, sir.
Michelle, and thanks, Dr. Mini. Thank you.
Thank you so much for making us a part of this.
Yes.
I would really hope that this was beneficial for each and everyone who attended it.
Certainly, madam. The insights were very good, wonderful. Especially the insights as compared to oxytocin in your trial. And as even in you all said that the even in the cesarean section, the drug is working better than oxytocin. That itself gives you confidence.
Perhaps we may come up with another study.
Yes.
With cesarean sections later on in time.
Correct. Right, madam. Thank you so much. Signing off.
Thank you so
Thank you.
much.
Thank you, sir. Thank you.
Thanks, Priyanka. Thanks, Maheshwari.
Thank you, ma'am.
Thank you, sir.
Thank you, Priyanka. Thank you, Hetero.
Thank you, ma'am.
Thank you, Priyanka.